All right. Hi, everybody. Welcome. Thank you for coming today. It is a pleasure to introduce Dr. André Green, who's visiting us from the University of Michigan. Drove down yesterday, told him to take 24, so he had a very pleasant drive. I'm very excited to have him here today. I've followed his work since at least 2014 on insulin signaling for software, cool paper. So I'm great. It's kind of cool now to see him working on a Monarch system. I'm thrilled to find out what he's working on and where he's going to go with this research. So thanks so much for being here, and I'll let you take off.
Thanks, Brock. Thanks for the invitation. Thank you all for coming. Let's just jump into it. So I titled this talk, Making a Migratory Monarch, very cryptic, because it kind of exemplifies how I think about the approach to studying monarchs. For a second, I thought that I wanted to be an engineer. So took a few of those courses, but realized I was kind of missing my kind of big picture curiosity questions. But what I'm still interested in is mechanism, understanding at a pretty deep level how something works. And with the monarchs, which we're going to talk about, kind of why they're so fascinating in a second, what I find kind of most troubling, in a sense, for myself is we're not yet able to basically create a migratory monarch in the lab. Despite knowing how other kind of migratory systems are induced, behaviors are induced in other systems, we can't do it with monarchs yet. So for me, that's kind of one of the primary goals, I'd say, of this first kind of stage of my research, is understanding enough about how monarch migration works such that I can kind of recreate the essential components in the lab. So with that intro, thinking about monarch migrations with respect to other insect migrations, why are we so focused on monarchs? So it does turn out that monarch migration is unique compared to other insect migrations. I have two examples here. So in this bottom left, we have the painted ladies, which also undergo kind of these continental scale movements, but they do it over several different generations. So different generations of individuals will make these continental scale movements. And then in this bottom right corner, you have the green darner dragonflies. They also move on a continental scale, but what's different here, or different about monarchs, is that monarchs, one individual will move in both directions. So they'll move bidirectionally, and monarchs actually have to have very precise navigation because they end up in very specific places, which we'll talk about. So that must mean that they have specific navigational capabilities, and no individual makes that trip more than once. So that must mean that they do it naively, and it must be programmed in some way. So to give you an overview of the monarch migration, so in spring and summer, you'll see monarchs spread across North America. So these dots are actually from a citizen science website called Journey North, where each dot is representing the sighting of a single individual butterfly. They're kind of spread across this range in spring and summer. They're reproductive during this time. They'll live a few weeks long. They are mostly looking for food and mates at this time. And that will occur over 2 to 4, depending on weather conditions, overlapping or partially overlapping generations. And in this bottom right corner is their major host plant, milkweed, Stephanus stevia. But of course, in fall, something dramatically different happens where these individuals, instead of looking for mates, they will actually divert their resources from kind of reproductive development to storing as lipid resources, because they're going to make an up to 3,000-mile flight to these very specific locations in central and southern Mexico. So these photos are just showing showing what those dense clusters look like, because you take all of these individuals from across North America and you condense them to about the size of 5 football fields altogether. So that's what this ends up looking like. And it's described, I think, like in a lot of popular press as whenever you're looking at these trees, and I mean, this is an adaptation, that it looks like just dead leaves on these branches because they're being weighed down. But then you'll see kind of this flicker of orange because one has opened its wings, and then you realize you're actually looking at thousands, millions of individual butterflies on these branches. It's pretty remarkable. So what these data show are, these are also Journey North citizen science records, but instead of each dot representing an individual butterfly sighting, these are actually locations where clusters of monarchs have come together at night, because monarchs will fly individually during the day, but then group together at night in hundreds to thousands of individuals in what are called roosts. And these are the locations of those different roosts. And you can see by the increasingly dark color, they're kind of funneling themselves towards their final overwintering sites in Mexico. Now, they'll spend the entire winter in Mexico, and in the subsequent spring, that's when they'll mate, and they'll start to make their way back northwards. But they'll only kind of reach mostly kind of the southern US, maybe up into Oklahoma as well, because they're looking for the appearance of their host plant milkweed, and it's starting to appear as it's getting warmer in that northern range. So that means that these single individuals are coming from this northern range, coming down to Mexico, and starting to go back into the southern range in the subsequent spring. So the central question that the lab that I'm interested in with the lab is, how exactly does something like this evolve? And we approach this from several different angles in the lab, but I'm going to focus on this kind of last kind of set of questions. So we think about how migration evolves de novo in cases. How are there rapid evolutionary phenotypic transitions? In the last few slides, I'll talk a bit more about those rapid transitions. But I'm going to focus in this talk on thinking about what exactly makes a migrant, or kind of what actually is novel about monarch migration. So the two more specific questions that I'm going to think about in terms of navigational mechanisms, what navigational mechanisms are they utilizing on their trips? And in terms of that bidirectionality, which makes monarchs unique, we're going to think about how do monarchs know that they made it to the right place? And what does it mean to be in the right place? So first, for the navigational mechanisms, kind of the framework that I tend to think about this is the Morton long distance navigation hypothesis. And the basic idea is that a long distance trip can be broken down into these 3 different phases of the trip. There's an initial long distance phase where you just need to go some broad direction that's south or southwest for monarchs. There's this second kind of middle phase or an intermediate phase called the homing phase. You're broadly in the area of where you're going to specifically end up. And then finally, you're at a pinpointing phase. Once you're in a broad area, how do I get to the specific set of trees that we're interested in? Because monarchs will not only return to go to these very specific sites in central Mexico, they'll return to the exact same sites year after year, even the exact same trees year after year. So as a part of the Morton hypothesis, the idea is that different sensory cues are being used for these different phases of navigation. So whereas maybe in the long distance phase, there's a stronger focus on vision and potentially magnetoreception. Once you get closer to the target, there's kind of a transition or switch into focusing on olfactory cues, as an example. Well, we do know a few kind of interesting things about how monarchs are making, how monarchs are navigating, particularly in this long distance space. So we know that they use the sun as a compass, and it's a time compensated compass. Based on the number of hours of kind of light that they've had, they'll change their direction to make sure that they're going in a particular south-southwest direction. So the kind of primary experiment that's been used to do this is called the Morse Frost Flight Simulator. Looks like this, and basically tether a monarch such that it can only spin around. And whenever you do this experiment in spring or summer, individuals will either just kind of spin around in this apparatus, or if they do choose a direction kind of as a group, it's not a coherent direction. They're just kind of choosing random directions. Whereas if you perform this experiment in the fall, you have individuals that are kind of flying directionally in a south or southwest direction. So kind of that's one of our cues into how are they getting to these specific sites. And showing that the kind of sun is specifically required for this on cloudy days, you lose that kind of southwest directionality. We also know that monarchs do use magnetoreception as basically a secondary system for navigating on, and presumably on, using the system on cloudy days. And it's shown through some actually pretty nice and elegant experiments from the Merlin lab that we know some of the protein players in this cryptochrome is a light sensing protein that's required for the monarch magnetoreception, which is a major point that I want to hear. Okay, so how we are extending this information that we know about how monarchs navigate is that we are attempting to track individual monarch butterflies across the migration. So the idea is that we'd be able to put these tiny environmental-based geolocators on them somewhere in their northern range, let them go. And we'd be able to recover kind of signals from them in Mexico. I'll explain. Well, this has been a collaboration with, well, these are the main PIs who've been working on this work, including myself. And how this system works, I mentioned it's an environmental-based geolocator. It can sense light and can sense temperature, and we're adding the capability to sense pressure. But the idea is that if we know the length of day and the time of sunrise and sunset, that's going to be deterministic of your location on the planet. So this sensor, kind of strapped on the back of monarchs, will collect that information. It will store it along their trip, and then once the butterflies get to Mexico, we just need to be within certain range, and then we'll be able to remotely download the light and temp data that they've collected. And then we can use our localization algorithm to then back out what the locations were along the trip. Okay. So this is, this is the basic idea of what we are trying to do then. We'll tag them up here, well, somewhere in their northern range. Of course, the goal will eventually be in Michigan, but we've just done a pilot where we've tagged them in San Antonio, and they'll fly down. We are going to meet them in Mexico in December. And right now we need to be within half a kilometer of them. Well, we think we can get up to half a kilometer. Actually, we have to be within 300 meters at this point. But because of how densely packed individuals are, that can mean covering millions of individuals within those 300 meters. And in order to start to infer kind of navigational mechanisms, the idea is to identify from those paths, specific zones that correspond to the 3 different phases of the navigational task, and either use gene expression or imaging techniques in order to infer what sensory cues are important in those different zones. So one aspect of this work that I've really enjoyed is the fact that in order to basically train localization algorithms, we've been collecting real world data from the public. So what we've done is basically sent large versions of our sensors to volunteers across, so we have volunteers in the US, Canada, and Mexico now. And we just ask folks to place them in their backyards. We have a few kind of instances of dogs eating them, but most of the time we get the data back. But this allows us to understand kind of some of because the hard part about the localization is going to be kind of the noisiness of the data. So shadows from trees, just lots of different things can make the data a little bit messy. But from what we've been able to kind of achieve so far in terms of better localization, particularly in combining light information and temperature information, we've been able to get what I think is pretty astounding resolution given the system in terms of being able to get better localization for individuals. So in the order of kind of tens of kilometers, which is, I think, a useful stat. So in order to get a little bit of proof of principle with the system, last year we attempted what ultimately, in the larger scheme of things, was a helpful experiment, but maybe considered an initial fail. But here's the idea. I thought it was a cool idea for the experiment. I mentioned that monarchs will normally migrate as individuals during the day, but folks always bring up the point to me, oh, we've seen these photos of just tons of monarchs kind of in the swarm that are flying about. Now, that is likely because what monarchs will do is once they reach large geographical barriers, like Lake Erie, for instance, they will congregate at strategic locations. So this particular strategic location is Point Pelee in Ontario. They will wait for the right weather conditions, wind conditions in particular. And once they have those conditions, they'll basically kind of island hop over that barrier. So this is the northern Ohio shore here. So because our system requires that we know where the monarchs are going, normally during fall migration, until you reach Mexico, that's a really hard thing to do for any single individual. But with these individuals, we have a better idea of where they're going. So to help us to tag up here, recover a signal from individuals down here. Well, we actually we weren't able to recover any signals down here. It turns out that our timing was off, so they weren't traveling as a large group, which means that they were kind of dispersed everywhere. But it turns out that there was an individual who was just kind of flying around Ontario. The weather conditions were also not great. It was a bit chilly and winds were in the wrong direction. So this individual actually hung around for a little while, but I was actually able a few days later to actually send out a ping for any individuals in the area. And I got a signal from this bug here. So that already kind of was huge for us because it meant that, okay, well, we can detect signals from individuals that are in the area. We're able to successfully download all of its data. We got an idea of how long that would take. And we were able to use the data from that individual and actually really accurately localize it to this point in Ontario. So that was really hopeful for the pilot that we set up this year, which was basically giving them a shorter distance over which to kind of make the trip. So I tagged in San Antonio. We'll recover Mexico this December. So, and actually I should note that we very much so kind of recognize it's still a needle in a haystack problem in terms of kind of recovering signals from these individuals. But that's where the fact that we don't have to physically recover the individuals kind of comes into play here. And that distance over which we're able to remotely download the data is critical. So we are still hopeful that with even kind of a few hundred individuals tagged, that we'd be able to get a small enough number of individuals in order to be able to say something biologically meaningful, just in addition to kind of demonstrating the kind of technical task. So for this second question of thinking about bidirectionality and how monarchs are getting to the right place. I mentioned before that these locations that monarchs will end up, and it's not kind of one single spot, but they are somewhat spread out. But these locations are highly regular, so much so that there's strong ecotourism that's built around these sites because they know that they're going to be there. In order to try to understand kind of behavior at these sites and how it may be changing, what we spent kind of a lot of the initial time in building the lab is coming up with new tools for assessing flight directionality, things that are easier to work with, a bit more just facile broadly. And in addition to being a bit more facile, making them much less invasive, such that we can capture individuals, test them, and then release them if and when necessary. The fact that and this also is based on a lot of work that's come out of Patrick Guerra's lab, actually just put a Band-Aid on monarch specs once you prepare them. And this allows you to kind of test individuals multiply so you can understand what's happening either over time or after different conditions or in different conditions or after different manipulations. There is a higher efficiency of testing in this particular system. Basically, any butterfly that we will put in here, most of them will fly, but even we consider non-flight to be important and useful data. Whereas in the former system, only maybe kind of 25 or 30% of the individuals that you put in turned into useful data, which is not bad for kind of behavioral experiments, but it just means that you're using a lot more butterflies than really need to. We're able to measure some other interesting parameters a lot more easily in this system. For instance, we can measure kind of wing beat frequency pretty easily by the changing size of the white box. But we knew immediately that the data would be a lot noisier because the monarchs have a lot more freedom to do things in this assay. So we need to just kind of make sure for ourselves that the assay works. We recapitulated some of the things that we know, how it should work. So we have summer non-migrants on the left, fall migrants on the right. And in these plots, these directional plots, each circle represents an individual butterfly. The size of the circle represents how long it kind of flew in our assay. So we just recorded them for 15 minutes. And larger circles indicate that they flew for a longer period of those 15 minutes, fraction of that 15 minutes. So as expected in fall migrants, you see, well, first of all, you can notice that individuals seem to be flying for a longer amount of time in the assay, and their direction is broadly to the southwest, although there are certainly there's variability there, which we saw from previous experiments, and that was expected. Whereas in summer, there's no overall kind of group directionality there, and they're flying for kind of less time in the simulator. Now, this bottom row represents tests done in cloudy conditions. And as expected, summer individuals aren't directional in cloudy conditions, and they were barely willing to fly for us, where in fall, individuals were still willing to fly for us, which that we know is true, but they're no longer kind of directional to the southwest. So some initial checks are kind of working out in the system, in the assay. So the data on the left are just reiterating the fact that in summer, this is percent flying time on the y-axis. In summer, individuals are less willing to fly, versus in fall, you have individuals that are much more willing to fly. But on the right, what we actually show is that for those individuals that are strongly directional, which is measured by a modified R or Rayleigh value, they have a preferred direction that tends to be southwest, which checks out with what we'd expect for these for fall migrants. Given that we found this difference and kind of flying time between fall migrants and summer non-migrants, we basically used an unbiased clustering algorithm to actually classify individuals as sustained flyers or trivial or non-flyers, and kind of you pull out that difference that you have a lot more sustained flyers in fall versus summer, which is again what we'd expect. But then kind of the real question that we're after here is, well, so how do monarchs behave how do they behave in Mexico? What changes, if anything? And it turns out that they do change it, so maybe as you'd expect. So you have a much smaller proportion of sustained flyers once they get to Mexico. So that means that they're kind of no longer strongly motivated to show migratory flight. And because of that, they kind of lose that southwestward group orientation that they have as fall migrants. But something also interesting that came out of this was, now if we go back to, and I didn't explain the colors the first time that I showed this. The colors are actually given the time of day that we performed the test, going from about 9 or 10 a.m. To about 5 p.m. Actually, so I didn't point out before, but in the summer, you can notice that there is a time of day dependence on the direction in which they're flying, and actually that makes a lot of sense. They're initially flying early in the morning, they're flying towards the east. Later in the morning, later in the day, they're flying towards the west. If you just release the Monarch into a room, they're going to fly towards the brightest light source. So this is them tracking the sun from east to west, kind of during the day. Now, in fall migrants, you don't see that signature because they're always looking to get towards the southwest, no matter the time of day. Overwintering individuals, though, also show a time of day dependence in the direction that they're flying. And it seems to be kind of axial from southeast to northwest. Now, interestingly, and this is something that I'll have to follow up on this upcoming season, the individuals that were collected for this experiment, where they were kind of taken from, the actual cluster that they were in was northwest of kind of the testing site that I was at. And the testing site was about half a kilometer away from the actual cluster of individuals. But what this broadly corresponds with is that if you go to Mexico, there you can absolutely see these daily movements of hundreds to thousands of individuals throughout the day. And it's thought that they're either kind of either moving back and forth towards water sources. It's not exactly known why, but we know that these daily movements will occur at the overwintering sites. Now, the fact that monarchs stop at these specific sites in Mexico may not be super surprising. Well, we know that they stop there, so maybe it's not super surprising that behavior changes there. But what we also knew from work about 10 years ago is that we've talked about monarchs needing to go back northwards in the spring. In order to do that, they basically flip their orientation from southwest to northeast in the spring. And it's some kind of a few weeks of cold that's specifically required to do this. It actually turns out that photoperiod doesn't matter in order for this flip to occur. And also what's really interesting about this is that if you took fall migrants and you just held them in kind of a simulated fall chamber for a few months and then tested them again, they continue to fly in a southward direction. So it means that they need that cold signature or cold signal in order to flip their directionality. So with this information about remigrants, it actually kind of turns into this model of fall migrants actually being pretty dynamic in the sense that they are initially these compass-oriented individuals who are flying southwest, kind of with the sustained flight behavior. In spring, they flip that orientation to the northeast, but there has to be this period in the middle where they're no longer compass-oriented, but they are flying on this particular axis depending on the time of day, and they're performing much more kind of local, non-sustained flights than sustained flights. So altogether, I put it together kind of in this model of there being kind of 4 dynamic or environmentally dependent switches that monarchs have to undergo in order to kind of achieve the bidirectionality component of their migration. First, of course, there's seasonal plasticity in that in summer you're non-migratory, so you are kind of dependent, time of day dependent in your flight because you're just following the brightest the sun. And then you're performing these local flights that aren't sustained. But in fall, now you have this much more kind of dynamic behavior that changes depending on either environment or location. And that's what we still need to work out. The second and third switches are kind of dynamic orientation, whether or not you are following a compass or whether or not you are just following or you're determining direction based on the sun. We know in migratory individuals that direction itself that you follow will change from southwest to northeast in the spring. And then there's this propensity to fly, or you just kind of a local flyer that isn't looking for kind of super sustained flight, or you're a more migratory like sustained flyer. And kind of why I separate out these 4 different switches, because I ultimately come back to mechanism. So now I have kind of 4 specific kind of phenotypes that I can look for to try to understand kind of how exactly is each one being controlled. And we have some initial ideas about the 4 of these just from experiments that have been done with monarchs over the past several decades. So we think that there might that seasonal plasticity is likely to be developmentally controlled, and there are some kind of potentially really interesting differences in brain plasticity across seasons that we're starting to look into. We know that there's an interplay between antennal and brain circadian clocks that control orientation in monarchs. In terms of flying either in a sustained way or not, we know that juvenile hormone has been linked to controlling strength of flight in different insect and particularly migratory systems. And all that we know so far about how kind of direction flips is kind of this cold requirement, but understanding how this cold interacts likely with this with these brain clock interactions is still kind of open. But this is work that's not only being done kind of in iLab, but across a few different Monarch groups. Okay. So after all of that description, I actually, I haven't really gotten into the why are they going to kind of, how do they know they're in the exact right place? Whereas in other systems, in salmon or sea turtles, they can use different forms of habitat imprinting. So either using geomagnetic or olfactory cues, or there can be systems for natal habitat preference induction. Basically, where you're born, there are some cues that you initially pick up that you then have a reference for returning to later on. But of course, that can't be possible for monarchs because they've never been to these sites before, or some of these systems use different older individuals will teach younger individuals for learning, and of course that can't be possible for monarchs as well. The current best hypothesis for why monarchs are going to particular places is that there's some particular microclimate that's special about these particular kind of trees and OEML forests in Mexico. But the kind of what's really interesting about that is in this particular figure here, this peach area is where you'll find where the monarchs will overwinter. Yet this green area is showing where there are OEML fir forests. These forests absolutely occur kind of throughout the range in that area, but monarchs are coming here. So we're still trying to figure out, well, why here versus these? But what this behavioral experiment now then allows us to do is we can basically bring the simulator to Mexico, test in different areas around here to understand how behavior either changes or is different in locations around, to understand, okay, will they orient towards here? Do they need to be kind of switched at some point? We now have just an experiment that allows us to kind of answer some questions. So I don't have an answer, but I think I have a method to get to it. A point that I wanted to make that's relevant to monarchs finding out where they're going is the fact that kind of broadly, monarchs, their populations are highly dynamic and in flux. So these are the data that I think a lot of people tend to be concerned with the popular press. The fact that 20, 25 years ago, you had these really large numbers of monarchs that were making migrations to central Mexico, and that number has kind of pretty significantly declined up to this point. And we know that kind of there are many different factors responsible for this dynamism, and particularly climate or weather factors, particularly in the breeding range, but also loss of breeding habitat more broadly, disturbance at their overwintering sites, infections. Protozoan parasite is kind of particularly nasty for monarchs, and also potentially insecticide use across the breeding range. But I think one of the kind of major points that I have started to kind of really recognize about monarchs, despite kind of the more dire kind of conservation stories that come out about them, is actually that they're pretty resilient. And I say that, even though kind of showing you these data, because it turns out that their overwintering sites and likely their navigation routes seem to be diversifying. So I mentioned that monarchs are found in this peach area. Maybe 10 years ago, monarchs were starting to be found in a few of these sites in this blue range here. And it was estimated that there were 1 million individuals that formed kind of a colony there. So it is a maybe 0.5 to 1% of the total population, but it's a significant group. And there are other indications that monarchs are basically starting to spend their winters in other places. As well, I've been involved in some collaborative work with potentially investigating that monarchs are using this eastern migration route through Florida, Cuba, and into the Yucatan Peninsula. How old this might be, or kind of what proportion of the population might be doing this, it's likely super small, but an indication that they have the ability to basically change these things that we thought were kind of immutable aspects of their migration. And lastly, kind of getting back to the central question of the evolution of migration is what I'm ultimately interested in. How we broadly do this is we use the fact that actually monarchs are a global species, and most of the populations that are found across the globe don't migrate. So each of these dots represents prominent populations of monarchs. And monarchs were initially or ancestrally migratory in North America, but there have been 3 kind of major dispersals represented in these different colors: a Pacific dispersal in blue, a Caribbean, Central, and South American dispersal in red, and an Atlantic dispersal in purple. And it's only in North America and in Australia and New Zealand that you'll find migratory populations. And it's presumed that it's in these locations because there's enough environmental variability that will then kind of necessitate migration. Because in these other areas, you have year-round persistence of milkweed, such that there's no strong incentive for migration at this point. So we then kind of do comparative work in which we'll perform kind of the straightforward common garden experiments with migratory and these resident populations in order to kind of understand genetic versus environmental contribution to different phenotypes among these different populations. And there's already been, I guess, an initial population genomic scan that you might expect in order to begin to understand genetic basis of differences between resident and migratory populations. And the kind of the focal story whenever this paper first came out, kind of you have this nice, what I call like the nature peak, that comes out at this gene that's called collagen type 4, and a basement membrane component protein that controls or is thought to control flight energetics. It turns out that this or an allele of this particular gene in the migratory population allows flight respirometry to just be much more efficient than in resident populations. So this then kind of being the go-to gene for explaining how monarchs are able to make such a long distance migration. But also in this scan, it turns out that kind of 2% of the genome showed up as highly divergent between migratory and resident populations. Of course, a lot of that probably wasn't responsible for the divergence, but then subsequently occurred. But what that means, though, is that there's kind of a large, kind of high, well-distributed adaptive range, essentially, for genes that might be kind of responsible for creating difference between residents and migrants. And it's those kind of 4 different the dynamic switches that we kind of identified before, looking for kind of what of the genes that have come out as divergent may be kind of related to those functions that we identified as being important for dynamic migratory flight. I guess. And so what I've loved about this work is that it has involved kind of so many people, and it seems to kind of grow by the month. With respect to the work that came from the lab, a lot of it came from a technician, Sean Polidori, in the lab. But with respect to the tracking project, I mentioned it's a collaboration between 5 different PIs and now I think over 300 volunteers now who contributed data for us. And we've been super lucky so far to be funded by a few different folks here. And with that, I'll take any questions that you might have.
So with the Pacific population of monarchs, have they adapted to have, I don't really know the climate there that well, but have they adapted to have a south-north as opposed to a north-to-south migration pattern?
Mm-hmm. So I kind of glazed over the fact that there are these monarchs on the west side of the Rockies that also perform a shorter scale migration than the Eastern North American population. They will, so their migration will be kind of like northeast to southwest. In the spring, they'll migrate out kind of northeast, and the fall, they will migrate to sites along the Pacific coast and central Southern California and northern Baja Mexico. The distance is a lot shorter, or that's one of the kind of primary differences in the Western population versus the Eastern population. But you know, whenever we think about them being adapted to do so, that becomes kind of a tricky consideration, because it was recently found that if you sequence Western and Eastern, their genomes are basically indistinguishable. Yet they have these two majorly different behaviors, suggesting that they are interbreeding somewhere likely Mexico. And that means that in Mexico, you have some type of split of individuals, some going back to the northeastern range, but then some going northwest to back towards Mexico west of the Rockies. We are still kind of totally in the dark about how that's happening, or how that's happening.
So you mentioned that they navigate both using solar cycle and probably magnetism.
Mm-hmm.
Has anybody looked as a natural experiment in declination, so the difference between magnetic north and celestial north as it changes across the continent, to see like which of those they use more heavily, or if they integrate the two, or if you take, for instance, like a monarch from the East Coast to the West Coast, will it not navigate as well because the declination is different?
Aha. So that level of understanding the magnetic compass has not been done yet, but there's been, I'll say, one major translocation experiment that's been done. I believe it was from Ontario to, I don't know if it was as far north as British Columbia, but there was a major westward kind of translocation. And that was an initial suggestion that monarchs potentially were using a vector strategy, because in Ontario, you expect a southwestward direction, and westward, you expect, if they're trying to go to Mexico, they're going to switch to southeast. They continue to go southwest. Now, I mean, actually, after just talking about kind of what happens with the Western population, what they weren't kind of able to disentangle is kind of, were these individuals genetically kind of going to, wouldn't be genetics because the genetics aren't different. Or it'd have to be some either expression or epigenetic signal that potentially could differentiate kind of what direction should you be going. But at least in that experiment, they continued that southwest direction, suggesting that they're using a vector navigation strategy that they weren't able to compensate for the change in location. Some folks had an issue with the experiment because they were saying monarchs were in a place that they would not normally be, or that the nature of the translocation kind of wasn't natural, allowing them sufficient time to kind of recover from it. I'm not sure if I completely am on board with the kind of they're in a place they've never been, so they couldn't kind of figure it out. One would think that if there is some kind of map-based compass and they do use either different geographical cues or some map-based sense, that's kind of the definition of what, if you're in a place that's unfamiliar, you kind of reorient. But in terms of how much time does it take to reestablish kind of where you are in order to maybe remap yourself, that might be maybe a contention to that experiment that I might buy. But none of that was done with respect to thinking about changing magnetic conditions.
So if you need this whole snap down in Mitchell Pond to reorient the compass, how might that be affected with climate change and not having a full snap?
Yeah, that is one of the primary questions that we are looking to get work, collaborative work to understand. One of the things about whenever monarchs reach their overwintering sites is actually whenever they first get there, they're actually quite dynamic. They're moving around from kind of initial site to site before they choose these kind of really common locations and stay there. And part of kind of one of my suspicions that's happening is they are almost kind of testing for kind of the right conditions, the right locations, temperature being one of these really important variables, because we not only know that it has kind of this directional control and needing kind of sustained temperature for a while, but also in thinking about surviving winter, because they've entered diapause, they need to go through kind of those lipid resources that's at a particular rate such that they can survive the entire winter, but not so quickly that they become much more susceptible to infections if it warms up too early or something. One might expect, because of particularly the fragmentation that's happening at the overwintering sites, it hasn't been demonstrated that that's causing necessarily warming kind of at the sites, but kind of broadly, the area is kind of warming, but you find the monarchs are actually moving higher and higher in elevation in order to get that optimal temperature component. So you might expect then that once they're no longer able to move farther up in elevation, then that's going to be will be suspected to be pretty dramatic. But I also think that's where this kind of behavioral these potential behavioral adaptations of monarchs choosing different sites becomes much more of an important consideration, that maybe in a sense like there's writing on the wall that these sites are becoming a little more dynamic, a little less stable than they once were. So if they're able to identify kind of other sites that provide what they need, they're going to those sites. So what I think warming climate may do then is probably even further reduce monarchs showing up in these places that we've kind of seen them show up for so long, which I mean, for them, it's great. I mean, for the local economies that are based on kind of monarchs showing up there, it's not great.
Kind of being into that, is there any historical documentation or evidence that there used to be a much larger range for the host plants, and that there was a correlation of the population
Absolutely.
spread out? So there is documentation.
Absolutely.
What is the scale of the map that you showed with the green and pink
Ah.
areas? Like how far the distance are those?
What are we talking about? So we are talking about, this is going to be maybe on the order of 400 kilometers. This is Mexico City here. This is about 2.5 hours drive. Yeah, so with 200 kilometers or so. So yeah, I'd say on the order of about 400 kilometers.
Do you think it's possible? I know you showed a lot of the non-migratory populations. The one that always stands out to me is Florida. Is it possible to retrain them to become migratory?
This has become, so this has been one of the big questions, right? So there are a lot of monarchs in a lot of different places. Can they become, can you make them migratory? So in addition to, because I focused on behavior, other folks have studied other components that make the migration successful, for instance, going into diapause. Resident populations they will go into diapause. Now it is quantitatively different from migratory populations, but it is possible. But what we do also know is that after relatively short, a few generations of inbreeding, and this is work that was done with captive bred individuals from actually monarch breeders in the US. After just a few generations, they lose some of this directional kind of flight ability. But I mean, that would suggest with some potentially some kind of outbreeding between the two populations that you'd be able to recover kind of that behavior if it were selected for. And that strategy, basically a translocation strategy for conservation, was one of kind of the, I'd say, well, maybe not the main impetus, but one of the considerations whenever monarchs were initially being considered as listed as threatened back in 2014, kind of could such a strategy work? So overall, there's certainly differences between kind of the residents, even in Florida and these other locations and in migratory. I don't think it's totally clear that these couldn't be kind of through certain breeding schemes kind of used to increase kind of a migratory monarch.
Is there behavior?
Yes, the behavior is totally different.
Migratory.
Yeah, so their behavior is totally different. It would have to be kind of a, it would have to be some type of, or I imagine it would have to be some type of crossbreeding program that the teaching would, I don't think that would work. But as a source, an initial source of individuals, I don't think that could be ruled out, I suppose.
I mean, yeah, if they go through 4 to 5 generations.
Right, so they do kind of, You don't have selection of migration over kind of a few generations in between, and we still don't. Kind of basically kind of studying kind of those temporal genetics hasn't been done. It would be super interesting to understand kind of what type of potential genetic bottlenecks are occurring over the course of a year.
Okay, we are up at the end of our time here. If you have more questions for Dr. Green and your grad student, you can ask those at lunch. If you have questions and you're not a grad student, I'm sure he'll take an email or chat.
Yeah, happy to.
So thanks again for your time.